Ich glaube, unser nächster Gast ist wohl die weit entfernteste Referentin. Ich möchte vor der Begrüßung zunächst einmal einen ganz großen Dank aussprechen. Ich möchte diesen Dank aussprechen an Rüdiger Stump, der zusammen mit einem Team hier wirklich es geschafft hat, diese Referenten zu gewinnen. Und auch bis nach Südafrika zu Frau Professor Dr. Resia Pretorius durchgerungen ist, die von der Stellenbosch University in Südafrika kommt. Sie ist weltweit anerkannte Expertin im Bereich der Hämostase und Entzündungsforschung. Ihre Arbeiten haben maßgeblich dazu beigetragen, das Verständnis über die Rolle von Mikrothromben und Thrombosen bei chronischen Erkrankungen wie ME/CFS und Long-Covid zu erweitern. Besonders hervorzuheben ist ihre Entdeckung, dass persistierende Mikrothromben eine Schlüsselrolle in der Pathophysiologie von Long-Covid spielen könnte. Im aufgezeichneten Vortrag mit dem Titel Thrombotische Endothelitis als Kennzeichen von Long-Covid präsentiert Professor Dr. Pretorius die neuesten Erkenntnisse zu thrombotischer Endothelitis als charakterischem Merkmal von Long-Covid. Sie geht dabei auf innovative Methoden ein, um amyloide Mikrothromben zu untersuchen und diskutiert, ob diese Mikrothromben tatsächlich als Blutgerinnsel betrachtet werden sollten. Zudem thematisiert sie die Präsenz dieser Mikrothromben in anderen entzündlichen Erkrankungen und erörtert, ob ihre Anwesenheit von klinischer Bedeutung sein könnte. Wir sind gespannt auf die tiefgreifenden Einblicke, die Professor Dr. Pretorius in die molekularen Mechanismen von Long-Covid geben wird und danken ihr herzlich, dass sie uns diesen Vortrag zur Verfügung gestellt hat.
Hello everyone. Thank you for inviting me to present our thoughts and work at this wonderful event. I'm so sad that I cannot join you in person today. I would have loved to meet you personally. Today I will be sharing our thoughts with you on thrombotic endotheliitis as a hallmark of long COVID. I wish to acknowledge a whole team of clinicians, research students, and collaborators. There are many more, and I will acknowledge them individually in the presentation where applicable. However, I need to acknowledge Doug Kell, my long-standing collaborator. We have been collaborators since 2011. Our lab's research will not have been possible if not for amazing funders like Polybio Research Foundation, and in particular Amy Proel, Balvi and Kanda Research Foundations. Also standing up to POTS, the South African National Research Foundation, and the South African Medical Research Council. We have studied platelet and erythrocyte changes in conditions like type 2 diabetes, Parkinson's disease, and Alzheimer's disease, hemochromatosis, and thrombotic stroke. These early insights revealed structural fragility, hyperactivation, and aberrant clot formation long before COVID-19. What we didn't fully grasp at the time was just how central these processes would become when a new global inflammatory challenge emerged. When SARS-CoV entered the picture, we were equipped with the tools and the insight to uncover something critical: the presence of persistent fibrinolytic resistant microclot complexes that stain with euflavin T, and how they structurally associate with immune triggers. This is the journey of seeing the unseen and making it count clinically. In the context of COVID-19, platelets are central in immune activation and general coagulation pathology, and they can form various complexes and obviously also within cells. This is known as platelet clumping. Panel A shows the traditional clotting cascade, where fibrinogen is enzymatically converted into fibrin through the intrinsic and the extrinsic pathways. This led to the formation of a fibrin clot with a well-organized structure, primarily composed of alpha helic regions. Panel B on the right illustrates the alternative pathological pathway we propose. In this scenario, circulating inflammatory molecules such as spike protein, bacterial components, other inflammagens such as serum amyloid A may directly bind to fibrinogen, where they may cause damage to the protein structure. Between 2014 and 2016, we discovered the potent inflammagen that comes from the walls of bacteria called LPS can directly bind to plasma proteins and cause them to become amyloid. The green signal that you see here is that of our marker of misfolded or amyloid protein, and it is called theophlavin T. Theophlavin T demarcates the misfolded amyloid areas, protein damaged areas of fibrinogen that has been damaged. On the left, you can see plasma from a healthy participant where theophlavin T is added and also thrombin to create an extensive fibrin network. In control samples, minimal green theophlavin T signal is seen. On the right in B, LPS was added in very low concentrations to control plasma, followed by thrombin. Significant amyloid formation was noted. This laid the foundation of our later work on acute COVID and long COVID. Here is an example of our earlier work on ischemic stroke. In the micrograph on the left in A is an example of blood plasma from a healthy individual with no visible amyloid protein misfolding. And the micrograph on the right shows plasma from a patient who had thrombotic ischemic stroke. Significant plasma protein misfolding is visible and demarcated by the green signal of theophlavin T. We also studied clotting pathologies in various inflammatory diseases by adding thrombin to the blood or blood plasma. Thrombin is the last step in the clotting cascade that transforms the soluble clotting protein fibrinogen to an insoluble gel or fibrin clot. It is a well-known lab method to study how fibrinogen clots into fibrin gel. In the micrograph in A, you can see a healthy fibrin net created with plasma of a healthy individual. In this healthy individual, this fibrin clot looks like a fishnet, but in diseases like diabetes, cardiovascular disease, or stroke, the fibrin clot becomes dense and matted, and this can be seen in the micrographs B and C. This might clog up the vasculature and can cause significant pathology. We were also interested in the structure of red blood cells and their death mechanisms turned eruptosis. On the left, you can see a red blood cell from a patient with severe iron overload disease known as hemochromatosis. Too much iron in circulation causes oxidative stress, resulting in significant inflammatory processes. On the right is a damaged red blood cell entrapped in fibrin from a patient with thrombotic stroke. In 2014, we were the first researchers to show that Parkinson's disease, there are significant red blood cell eruptosis. This we directly attributed to circulating inflammatory molecules. At that stage, the idea of the microbiome in blood was still a new concept. We argued that inflammagens like LPS from bacteria in circulation could also directly cause such pathologies. Now it is widely accepted that there is indeed a blood microbiome and that it has significant impact on pathology and disease progression. Fast forward to the pandemic. Early on in 2020, we looked at the blood clotting in acute COVID using the scanning electron microscope. We found that platelets were damaged and clumped together and attached to red blood cells. The fibrinogen and clotting proteins that are supposed to be soluble formed these spontaneous deposits. In the micrographs on the far left, you can see platelets that are hyperactivated. Note the yellow arrows. In the middle are micrographs where platelets are attached to red blood cells. Note the white arrows. On the far right, you will see micrographs of spontaneously formed plasma protein deposits that even glue together red blood cells. Note the blue arrows. End of 2020, reports from collaborators called Urgent Attention with a dire warning, persistent symptoms after acute COVID recovery, a condition called long COVID, first termed by Lisa Perego and the team of patient researchers. We identified the first cases in South Africa too soon afterwards. This was already end of 2020. Long COVID turned out to be a global catastrophe. It still is. Of course, we immediately sought to look at long COVID blood samples. Here you can see scanning electron microscopy micrographs showing red blood cells covered with microclots. We also used theoflavin T as a marker for our misfolded protein to identify these microclot complexes. Here is a close-up of the deposits on red blood cells where microclots seed onto intact cells with the potential to significantly impair their function. Turning to our proteomics research, in 2021, we published our first proteomics long COVID paper. As I mentioned, the term microclots has also originated from this first proteomics paper. I was interested in the protein footprints of acute and long COVID samples and compared them to healthy and diabetes samples pre-pandemic. To our surprise, we could not digest the plasma of the acute and long COVID patients with the laboratory standard trypsin digestion method. Our biochemist, Dr. Marie Vlok, developed a double digestion method, and we found numerous entrapped inflammatory molecules and also antibodies in the digested microclots. During the data analysis, we discovered, amongst others, alpha-2 antiplasmin, von Willebrand factor, complement proteins, and then also indeed various fibrinogen chains and coagulation factors that were entrapped in the solubilized fibrinolytic resistant microclots. In 2022, it became clear that we needed an easily accessible method that could be used in general pathology labs. We turned our attention to flow cytometry, but soon also realized that they will have to convince clinicians and researchers that microclots exist. So therefore, we needed to show them using an imaging flow cytometer. With the help of Balvi Research Foundation, PolyBio Research Foundation, kernels, and thousands of donations, we had the instrument within 6 months. 6 months later, we published our first imaging flow cytometry method with the hope that it would be a useful diagnostic method to quantify microclot complexes in plasma samples. So why is it important for us to determine if these complexes are present in circulation? We and others believe that these complexes may result in ischemia-reperfusion injury. So what exactly is ischemia-reperfusion injury? It is initiated by bursts of reactive oxygen species produced during the reoxygenation phase following hypoxia, and this is a well-known process in a variety of acute circumstances. We argue that this type of injury also underpins elements of the pathology of long COVID. Ischemia may be initiated via fibrin microclot deposits, thus temporarily blocking the capillaries. For instance, as exercise is started, reperfusion is a necessary corollary when it finishes. It is not only simply the size of the numbers of microclots present in healthy versus long COVID or any other disease with circulating inflammatory molecules, but their content, the inflammatory molecules of interest that might be entrapped inside them, their activity and their biochemical characteristics. Such characteristics make the concepts of microclot complexes ideal candidates for phenotypic characterization. Together with our pathology lab collaborators, Jan Kok and Felix Skolkman from Innes Laboratories in the University of Zurich, Alain Thierry from INSERM in France, and Doug Kell from University of Liverpool, we are closely looking at the phenotypes and the nomenclature of the term microclots. We have also been looking at the biochemical characteristics of the various molecules that us and others have found entrapped inside the fibrinolytic microclot complexes. We know that these microclot complexes stain with theoflavin T and also with amine trackers. However, inflammatory molecules associated and entrapped may also have their own amyloidogenicity. By using a website called Ameliogram, the amyloidogenicity of any protein can be determined. Here is an example of alpha-2 antiplasmin, the molecule that prevents clot breakdown. We have found this to be present inside these complexes using proteomics. In fact, almost all of the inflammatory molecules we have found associated with our microclot complexes have significant amyloidogenicity themselves, making them sticky and thus prone to attach to to fibrinogen and to other microclots as they roll over damaged endothelial layers. Using the amelogram site, such a diagram can be generated. On the x-axis is plotted the amino acid residue number, and on the y-axis is plotted the amyloidogenicity score, which is essentially the probability that a given sequence segment of a protein can form amyloid structures. Our latest thoughts suggest that there are various microclot complex phenotypes. Here are the possible microclot phenotypes we have identified so far, where the fibrinogen forms the main component of the little complex, resulting in a fibrinoid microclot, where fibrinoid microclots associate with nets or neutrophil extracellular traps, forming immune complexes, where this microclot form and seed onto cellular debris from damaged blood cells, blood vessels, and the membranes of these blood cells and blood vessels. This might also be a phenotype. There might also be a phenotype resulting in heterogeneous inflammatory molecule aggregates together with fibrinogen seeding onto the blood cells, endothelial cells that are still intact, but that might be themselves pro-inflammatory and damaged. Together with our pathology lab collaborators, we will now be looking closely at this type of phenotypes that we can find inside blood. We have previously added spike protein and LPS to purified fibrinogen and used the fibrinogen alpha chain antibody and our marker of amyloid protein, theoflavin T. Thus, this fibrinogen aggregating in the presence of LPS is one of our phenotypes. Many other of the inflammatory molecules like serum amyloid A and complement can also do that, including also spike protein. This can change our purified fibrinogen into a little insoluble structure that is THT positive. Bacterial biofilm can also be found in microclot complexes, albeit with very little signal. And we have long said that long COVID is a thrombotic endotheliitis. Here we have found endothelial damage molecules shed from the damaged vasculature that are associated with these microclots that stain positive for theoflavin T. Recently, we've also collaborated with Prof. Allard Thierry from INSERM in France. He is interested in net formation and neutrophil elastase, immunoperoxidase, and circulating DNA in long COVID versus control samples. We confirmed that nets are associated with microclots in long COVID patients. We used here cytostain, which is a red stain, and it's similar to Huscht, which is a very well-known DNA stain. Also theoflavin T for our misfolded protein areas inside these microclot complexes, and immunoperoxidase antibody, which is the blue areas that you can see in this complex. We have long said that long COVID is also a thrombotic endotheliitis and associated with red blood cell damage. Here we have found these cells just covered with microclots. As you can see, the red blood cells structurally associated with these complexes. One of the questions we frequently get is whether microclotting is related to macroclots as seen in thrombotic stroke. Circling back to our research many years ago, we found significant microclot deposits in blood samples from acute thrombotic ischemic stroke. This research was done in collaboration with a neurologist, Dr. Dame, in Pretoria. At that stage, we were more interested in the red blood cell pathologies and also did not really pay much attention to the plasma deposits. Now looking back at our data, the microclot complexes were hiding in plain sight. I'm happy to report with Doug Kell and a team of researchers and neurologists from the University of Liverpool and with Melanie Walker from University of Washington, we have published a paper in the journal Stroke showing that retrieved macroclots from patients that had a thrombotic stroke indeed contain significant amyloid proteins as demarcated with theoflavin T. This study was conducted in Liverpool and samples were collected under their ethics. This suggests that microclots are on the biochemical and physiological pathway to the formation of macroclots, as seen in stroke, suggesting that microclot complexes may even lead to macroclots under specific inflammatory and pathological conditions. We have just pre-printed a paper and it is now under review, done by a team of Stellenbosch researchers and my postgrad student Maxine Waters. We looked at the proteomic signatures of post-vaccine, post-infection syndrome that we call PVPIS and discussed insights into immune dysregulation and coagulopathies. We compared the protein signatures with previous published data from the proteome analysis of long COVID patients and found that individuals with post-vaccine, post-infection syndrome had distinct protein footprints, suggesting that vaccine injury might lead to a different physiological pathway of activation, but with the same symptoms. In this preprint with the Fahy group at Derby and under an MTA between the universities of Stellenbosch and Derby, we found that exercise may worsen symptoms in long COVID due to microclot fragmentation. The Derby team employed a 2-day regime of submaximal cardiopulmonary exercise testing called CPET, and they assessed 46 patients. We now know that many patients with long COVID report post-exertional symptoms that may worsen after physical and mental efforts. In this study, we attempted to explain why. We show that microclot fragment after exertion, potentially triggering inflammation and resulting in reduced oxygen transport. When fragmented, these complexes may also release inflammatory molecules that are entrapped inside of them into circulation, amplifying inflammation and impairing oxygen delivery. Our paper challenges the rationale for graded exercise therapy in long COVID. Most importantly, the effects of exercise should be objectively monitored, and patients with post-exertional malaise should be monitored even more carefully and then managed accordingly. We are also working with Jeroen den Dunnen, and we have optimized the microclot imaging flow cytometry tests in his laboratory in Amsterdam UMC in April this year. Jeroen will be using full-body PET scans to detect microclots in patient organs. This work is done under ethics by Jeroen's team in Holland. A team of radiologists from the University of Free State have recently found abnormalities in the carotid arteries that are atypical. This team is currently investigating the phenomenon more closely. You can see the fluffy deposit they have found and that they believe might be microclot complexes in the carotid arteries in this micrograph, and this micrograph is kindly provided by them. Note the red arrow. We have also been looking into the possibility to either isolate single microclot complexes or at least manipulate them with nanotweezers. Here you can see a video of preliminary results where we could drag along and even break off and move around the microclots with nanotweezers in the system. In our latest research, we looked at pseudoserum. Here is the depiction of the methodology we used. First, platelet-poor plasma samples was exposed to thrombin to form naive fibrin networks. Then the naive samples were subjected to thromboelastography analysis. The watery part remaining, the pseudoserum, that is devoid of all clotting factors and fibrinogen, was used in this study. It is thus supposed to contain only inflammatory markers in solution. The resulting pseudoserum was collected, exposed to thrombin to confirm the depletion of fibrinogen. Next, pseudoserum samples were incubated with purified, commercially available exogenous fibrinogen obtained from healthy donors and exposed to thrombin to form fibrin networks. Morphological analysis were conducted using light fluorescence and scanning electron microscopy. As mentioned, platelet-poor plasma samples were exposed to thrombin in order to assess naive fibrin networks of participants, figure A on the left. There is a clear morphological distinction between control and disease fibrinogen networks, with the latter showing enhanced density and disorganization, dense granular regions, different fibrin thickness, and anomalous deposits. The control fibrin clots, in contrast, exhibit structural uniformity, almost lack regions of granule densities, and exhibit notable pore sizes and spatially arranged networks. In Figure B, you can see micrographs of pseudoserum samples exposed to exogenous thrombin. The lack of fibrin networks indicates that all or most of the fibrinogen in platelet samples has been depleted in the TEG experiments. The last column represents the flavin T stained seroserum, show that there's an overlay in the patient cohorts. Some, but not all of the aggregates in the pseudoserum stain positive for theophlavin T. Here you can see in A micrographs of thrombin-induced fibrinogen networks formed from purified fibrinogen exposed to pseudoserum samples using light microscopy. In B, you can see micrographs of thrombin-induced fibrin networks formed from purified fibrinogen exposed to serum, and here we use scanning electron microscopy. Here we showed again that pseudoserum obtained post-TEG analysis and then this of plasma from two disease populations, long COVID and type 2 diabetes. If we expose these to purified fibrinogen, it results in notable aberrant fibrin characteristics, and that's similar fibrin abnormalities exist in naive plasma of samples exposed to thrombin. The ability of pseudoserum to drive these changes despite the absence of fibrinogen and other clotting proteins from the original patients emphasize that soluble molecules retained in the pseudoserum from patients can modulate the coagulation process on their own. Together with many researchers, we have also wrote a Delphi for long COVID. This modified Delphi electronic survey on long COVID was conducted under the governance and oversight of the World Health Network. Together with a team of clinicians, we also wanted to give some guidance to clinicians on how to determine if their patients indeed have thrombotic endotheliitis. We wrote a paper with title Vascular Pathogenesis in Acute and Long COVID, Current Insights and Therapeutic Outlook. It was published in Seminars in Thrombosis and Hemostasis in August in 2024, and it won the most popular paper for 2024. Here you can see Dr. Arno Krier, Dr. Dave Joffe, Dr. Graham Lloyd-Jones, Dr. Asad Khan, Dr. Spila Salomon, Dr. Jaco Loubser, Prof. David Apicino, and Prof. Doug Kell, as well as myself. To change outcomes in long COVID, we need to find measurable biomarkers. We must then stratify patients with precision and design clinical trials that are grounded in biological reality, not on assumptions. THG positive microclot complexes offer a powerful lens. They trap inflammatory molecules, reflect underlying pathology, and can be quantified before and after interventions. They may not only help us to define disease, they may also help us to define who responds to which treatment. Of course, introducing a new method and even a new term into clinical medicine is never easy. It requires robust evidence, relentless communication, and time. But the alternative is continuing to overlook a potentially key driver of disease. This is what seeing the unseen truly means, uncovering the invisible mechanisms that perpetuate illness and translating that knowledge into tools that serve patients. The journey from research to clinical utility is rarely linear, but with the right biomarkers, the path becomes visible. Here is our team. Every day we are working in our lab to find answers. Thank you for waiting for these answers patiently. Thank you very much. And I look forward to meeting you one day in person.